Hello, everyone. Hopefully you are doing well. That is so good. We are still continuing on our discussion about homeostasis, excretion, and osmoregulation. Last time we looked at the characteristics of homeostatic organs and the formation of tissue fluid. So how tissue fluid is being formed by the difference in the pressure at the arterial end of the capillary bed, how smaller materials are filtered out into the tissue, and then they again enter at the venular end. And we saw that if this tissue fluid is not picked up by the lymphatic system, then it results into a condition known as edema. So we proceed with our discussion, and today we are looking at the homeostatic role of the liver and the pancreas. Before you look at the homeostatic role of the liver and the pancreas, we shall first see the structure of the liver and the description of the microscopic structure of the liver. You can get more about it from Michael Roberts and Reyes et al. Advanced Biology or Biological Science, 3rd edition, page 667. When you look at the diagram, you are looking at the liver lobule, and once we extract one liver lobule from A, it appears in this format in B. You see it has the central vein, that is the intralobular vein, which is a branch of the hepatic vein. Then it has the liver cord, that is the chain of liver cells, the canaliculus, or you can call it bile. We have the sinusoids, where blood is flowing through, and then you have the bile duct down with the branch of the hepatic portal vein. No, it is the hepatic portal vein that brings blood from the alimentary canal. Much of the food that is absorbed from the alimentary canal enters the bloodstream through the hepatic portal vein, and then blood that leaves the liver goes through the hepatic artery. You can look, no, it goes through the hepatic vein, sorry, not the hepatic artery. So we say that the liver is composed of structural and functional units called the lobules, which are cylindrically shaped, numbering over 100,000, and each approximating to 1 millimeters in diameter. These hepatocytes, the liver cells, closely pack in each lobule in various rows radiating outwards from the center, and they are characterized by similarity in structure and function. They possess the prominent nuclei with the Golgi complex, and the peroxisomes with numerous mitochondria, the lysosomes, glycogen granules, and fat droplets. These peroxisomes contain catalase, an enzyme, and other oxidative enzymes that are responsible for detoxification. The liver carries out the role of detoxification. Then we also have the hepatocytes, which are in contact with blood vessels bearing microvilli. And these guys are numerous, the microvilli, to increase the surface area for the activities that will be done by the liver. Located between the lobules are triads consisting of a branch of hepatic artery, which brings oxygenated blood to the liver, branch of the hepatic portal vein, which brings nutrients from the gut, and the bile duct that drains bile from the liver, that is located between the lobules. Then the central vein, that is the branch of the hepatic vein, which runs longitudinally midway each lobe and is linked by sinusoids to the interlobular vessels, that is the hepatic artery and the hepatic portal vein. And these sinusoids radiate from the center to the periphery of the lobule, and their endothelial lining is perforated. So these sinusoids at the same time alternate with bile canaliculi. Those are the small canals which carry bile, and attached to the walls of the sinusoids are the microfungus cells known as the Kupffer cells. We shall see the role that is done by the Kupffer cells as we go on with the homeostatic function of the liver. Basically, that is the structure of the liver, of the microscopic liver cells. How is it related to its function? Because the liver performs a lot of functions, so it must have adaptations, the specific structures, the specific parts that this liver contains, or that is made of, that are responsible or that improve the working or the working action of this liver. So the Kupffer cells ingest worn out red blood cells. The bacteria fall in particles from the blood flowing through the liver. That is the role performed by the Kupffer cells. Then we also have the closeness of the hepatocytes with the sinusoids and the canaliculi. They are so close, the hepatocytes, with the sinusoids and the canaliculi, which enables them to receive nutrients and expel waste substances. Another adaptation, we look at the excellent blood supply, which provides nutrients to the cells and enables wastes to be carried away. So it has a very great significance to possess excellent blood supply, numerous blood capillaries, the ones that supply nutrients to the cells and the ones that transport waste products away from the liver. Now, when waste products accumulate, then they are affecting or they are bringing about or slowing down the working action of the liver. The hepatocytes, which are in contact with blood vessels, they bear numerous microvilli, which increase surface area for exchange of substances, as well as the liver is large, providing large surface area for metabolic reactions to occur. That is improving or increasing the surface area for metabolic reactions to occur. A lot of metabolic reactions are taking place in the liver. In fact, it is the most active metabolic organ, or it is the most active organ of the body. We have the hepatocytes bearing numerous peroxisomes, which contain enzyme catalase and other oxidative enzymes that are responsible for detoxification of poisonous substances in the liver. And its tissue is also elastic, that is enabling expansion to store large volumes of blood. A lot of blood is coming to the liver because it is the most, or it is the metabolic organ of the body. So you expect a lot of blood to come, bringing nutrients. So the liver or its tissue must be elastic such that it can accommodate these large volumes of blood. These hepatocytes are as well similar in structure, that is, they are undifferentiated, enabling them to perform the various metabolic functions. Those are the adaptations, and after looking at them, then we are going to see the homeostatic functions of this liver. What roles does the liver do, or how does it bring about maintaining a constant internal environment in the body? So we take our first stake about the homeostatic laws of the liver, and we are going to discuss about regulation of blood glucose. Now, glucose has very many significant roles, and the most significant role it performs in the body is it is being broken down, or it is metabolized to provide energy in the body. So it must always be maintained at a particular constant high level, and in the body it is maintained at a concentration of 90 to 100 milligrams of glucose per 100 centimeters cubed of blood, that is volume. That is approximately 0.1% or 0.1 gram per 1 centimeters cubed of blood. In spite of the fluctuations of up to a high 150 to 200 milligrams per 100 centimeters cubed, or lowering it up to 70 per 100 centimeters cubed. So sometimes it can go high up to 150 to 200, or it goes low to 70, and so on. But there is an interaction between the liver and the pancreas that enable the blood sugar level to be maintained at optimum concentration of 90 to 100 milligrams of glucose. And how does it perform that very role? Here we go. How the liver and pancreas interact to maintain glucose level constant. If there is a rise in blood glucose level above the norm, a condition or a situation known as hyperglycemia, it stimulates the beta cells of the islets of Langerhans in the pancreas to secrete the hormone known as insulin into blood. So that is what happens. If there is a rise above the norm, hyperglycemia, the beta cells are stimulated of the islets of Langerhans in the pancreas to secrete the hormone known as insulin into blood. Insulin binds to the body cells with insulin receptors and causes processes which reduce glucose concentration as the following. So there is an increase in concentration of glucose above the norm. So a hormone that brings about reduction of the glucose concentration is being produced by the beta cells of the islets of Langerhans, and this hormone is insulin, and does that role of reducing the glucose concentration by doing the following. That is, it increases cellular respiration in muscles and liver cells. That means more glucose is being broken down to generate ATP, forming carbon dioxide and water. There is increased glycogenesis, that is conversion of glycogen from glucose in the muscles and the liver cells. Glucose is being converted into glycogen for storage. There is increased conversion of glucose to non-carbohydrate substrates, such as the fats and proteins in the adipose tissue. And also, it increases the uptake of glucose in the muscle cells. So this restores the glucose levels back to norm. All these four ways, they are reducing the glucose from the high levels back to low normal levels. Now, if it happens and there is a sudden decrease in blood glucose level below the norm, a condition known as hypoglycemia, that means hypo means there is much water present and the glucose is little. There is inhibition to insulin secretion, but then there is stimulation of the alpha cells of the islets of Langerhans in the pancreas that will secrete a hormone known as glucagon into the blood. Being the only cells with glucagon receptors, glucagon binds to the liver cells, causing them to increase blood glucose level through the following. The other one was reducing blood glucose levels, whereas glucagon will be reducing the blood glucose levels. Through the following ways, there is increased glycogenolysis, that is hydrolysis of glycogen to glucose. We have increased formation of glucose from amino acids and glycerol. Those are the non-carbohydrate substrates. And there is also formation, or the process of formation of glucose from non-carbohydrate sources is known as gluconeogenesis. So insulin and glucagon, that is a note. They are not the only hormones that control the blood glucose concentrations. We also have other hormones that are present or that are always produced in such situations, such as adrenaline from the adrenal medulla, which causes hydrolysis of glycogen to glucose during acute stress or excitement. When you have a lot of stress or you are excited, adrenaline hormone is released because you need energy to maintain your stress or the excitement. So you must have glycogen getting converted to glucose such that it can be used for metabolism, to provide or to generate ATP. And thus, this increases and reduces its concentration in blood, respectively. We have another hormone known as cortisol from the adrenal cortex, which causes formation of glucose from amino acids and glycerol when glycogen exhausts or when glycogen stores are used up, hence increasing the glucose concentration in blood. Another hormone from the anterior pituitary gland is the growth hormone, which increases the glucose concentration in blood through fat breakdown. That is the growth hormone. Also, we have thyroxine, which stimulates metabolic rate, that is, increases glucose breakdown. So thyroxine performs the same role as insulin, and all these others, they are bringing about increase in glucose concentration. So these other ones are performing the same role as glucagon hormone. So it is not only insulin that is produced by the beta cells and glucagon produced by the alpha cells that are only involved in maintaining the blood glucose levels at 90 to 100 milligrams per 100 centimeters cubed of blood. Also, other hormones are involved. They also perform specific roles depending on the situation that an individual is in. If he's excited, he's preparing to fight, he needs a lot of energy. So such an individual expect adrenaline to do what? To increase being secreted by the adrenal medulla, which will bring about all those effects. Second note, In some people, there may be insufficient secretion of insulin, or the cells may be insensitive to insulin. Now, sometimes insulin is produced in low amounts, or the cells become insensitive to insulin. So if the cells become insensitive to insulin, that means its effect won't take place. You will have continuous accumulation of glucose. This results into a condition known as a diabetes mellitus. We have two types of diabetes mellitus: insulin-dependent diabetes caused by insufficient secretion of insulin, whereas we have insulin-independent diabetes, which results from insensitivity of the cells to insulin. So diabetes mellitus is a condition where you have high amounts or you have high blood sugars. A lot of glucose is present in blood, a condition known as hyperglycemia. Symptoms: you have high blood sugar, you realize you have diabetes mellitus. Those people you hear, they tell them that they are suffering from sugar. It is not sugar, but they are having high blood sugars in the bodies. We have glycosuria, that is presence of glucose in urine. You have frequent or fluent copious urination. Every time you have to go and urinate. You have abnormal thirst. You have visual disturbances, itching of the genitals. You have fatigue, rapid weight loss, drowsiness, skin disorders, itchy boils, and general weakness. All these are the symptoms of diabetes mellitus. So once you have one, you go and you get the doses of insulin, or you see if the body cells are the ones that are insensitive to insulin. That is when you have high amounts of glucose in the body. You can look at this diagram. It summarizes everything. It summarizes the control and regulation of glucose from the norm. If it goes up, you expect the detector and regulator, that is the pancreas, stimulus rise. It goes to the pancreas, those are the B cells, bringing about insulin that will bring about all those conversions from glucose to glycogen, increased cellular respiration, glucose to fat, that is within the liver, then in the muscles, glucose to glycogen, increased cellular respiration, and other cells, you have increased cellular respiration. So all that bring about a response which is falling glucose and restores it to the normal levels or the set point. If it is low, still we are seeing all these taking place. When it is low, you expect to have the effect of glucagon hormone, adrenaline by the adrenal medulla. Then you have the anterior pituitary gland, which stimulates the adrenal cortex to release the hormone cortisol. Then you have the thyroid gland releasing thyroxine hormone and also the growth hormone. All these bring about increase in glucose levels in blood, restoring it back to the norm. Some of the functions of the liver, out of the over 500 estimated, it performs. We have digestive, regulatory, and excretory. A, it maintains a steady blood glucose concentration by conversion of glucose to glycogen if it is above the norm. And if it is below the norm, that is vice versa, and under the influence of hormones. So the liver's carbohydrate metabolism involves the following, that is glycogenolysis promoted by insulin, glycogenolysis promoted by glucagon hormone, lactic acid metabolism initiated by the enzyme lactate dehydrogenase. Then we have gluconeogenesis promoted by cortisol and adrenaline hormones. That is gluconeogenesis, where we have glucose being produced from non-carbohydrate substrates. Those are the ways through which the liver's carbohydrate metabolism is being maintained. Another role of the liver, it regulates amino acids and proteins. That is, it is involved in protein metabolism in the body. In Roman 1, we say we cannot store excess amino acids in the body. So any surplus must be removed, and the liver performs this role through a process known as deamination. That is the removal of an amino group from the amino acid to form ammonia. That is the process of deamination. It is performed by the liver, where an amino group is removed from the amino acids to form ammonia, which in mammals is converted through a series of reactions. That is the Ornstein cycle. We shall look at that. And it is converted to urea, which is shed from the liver cells into the bloodstream. And from the bloodstream, it is transported to the kidney for excretion. As you know, urea is our main excretory product, which is released through urine. So it must be transported to the bloodstream and then to the kidneys for excretion. Once you have removed an amino group by deamination, then you have the amino acid residue, which is a keto acid, which is fed into the carbohydrate metabolism and oxidized to release energy, converted to glucose, glycogen, or fat, and stored. So the rest of the amino acid joins the carbohydrate metabolism. It is oxidized to release energy. Others that remain are converted into glucose, glycogen, or fat. Basically, the keto acid, depending on the amino acid and relative to the carbon atoms that they will be possessing, if it is a 3 carbon atom, a 4 or a 5, we expect them to join the Krebs cycle at different levels depending on the carbon compound present. You have citric acid, you have alpha ketoglutarate, that is the 5 carbon. Then when it also undergoes deamination, then you have what we call succinate and the rest. So depending on the carbon atoms present in a protein or a specific amino acid, that is where it joins the carbohydrate metabolism pathway. That is the respiratory pathway. The liver also performs the role or carries out transamination. That is the transfer of amino groups from amino acids to other organic compounds to form amino acids that are deficient in the diet. Because these amino acids are in large amounts. So once they are in large amounts, the only way we can remove them is that through deamination and transamination, transfer the amino groups from the amino acids to other organic compounds to form amino acids that are deficient in the diet. You can look at this one. It is the deamination where it is dependent on oxygen. It must take place in presence of oxygen. You remove an amino group, and then you have the rest of the keto acid, and then you form ammonia, which later, through a series of reactions, you are looking at it, it joins the Ornstein cycle. Ammonia combines with carbon dioxide to form urea. So urea is produced at the end of the day through the Ornstein cycle, and it is excreted through transportation to the bloodstream, and then it joins the kidney for excretion. A summary of the reactions of the Ornstein cycle. The amino group of an amino acid is removed and reacts with hydrogen to form ammonia. Then ammonia reacts with carbon dioxide to form carbamyl phosphate using energy from ATP. And the carbamyl phosphate reacts with the Ornstein to form citrulline. Reactions 2 and 3 occur in the mitochondria matrix, and the citrulline that is formed diffuses into the cytoplasm of the liver cells and reacts with aspartate to form adeninosynsate, and this one splits into arginine and fumarate. Fumarate enters the Krebs cycle because it is a 4-carbon compound, so it is resembling fumaric acid in the Krebs cycle after succinate. Then arginine that has been formed is hydrolyzed to form urea and ornithine, and then it is continuous. The ornithine will again react with the carbamylophosphate that is formed at the beginning when carbon dioxide reacts with carbon dioxide. Ammonia reacts with carbon dioxide to form carbamylophosphate using energy from ATP. So there is an addition of a phosphate group. That is why you have carbamylophosphate. Once urea has been formed, it is carried in the bloodstream to the kidneys for excretion in urine. The liver also performs another role of regulation of lipids, that is lipid metabolism in the body, where we say excess carbohydrate is converted to fat. You have stored fats are desaturated prior to oxidation. Then it synthesizes and degrades phospholipids and cholesterol. These guys should not accumulate in the body. Synthesizes lipid transporting globulins. And once there is excess cholesterol in blood, it is excreted into bile by the liver to avoid their accumulation, which can result into atherosclerosis, that is narrowing of the arteries. These cholesterol levels should not be allowed to accumulate, because once they accumulate, you expect narrowing of the arteries, which will result into high blood pressure, which we call atherosclerosis. Thus, this one causes thrombosis, that is blood clotting in the blood vessels. If cholesterol is greatly in excess in bile, it forms gallstones, which can block as well the bile duct. So cholesterol has a lot of side effects which are dangerous. So it shouldn't be allowed to accumulate in any large numbers in the body. That is how the liver regulates lipids or lipid metabolism in the body. Then this point which shows excess carbohydrate is converted to fat, that is wrong. Excess fats are converted into carbohydrates. That is conversion of fats to carbohydrates for oxidation. Then we have D. The liver forms red blood cells and lymphocytes in the fetus and breaks down worn out red blood cells in the adults. This is done by the liver's Kupffer cells. We looked at them, where they break down the worn out red blood cells to form bile pigments. And the pigment that is formed is bilirubin, which is excreted in bile. That is the bile pigment, bilirubin, and it is excreted in bile. Then you also have hemoglobin being broken down into globulin, a protein, and a heme group from which iron is removed and stored. When these guys, the red blood cells are worn out, this is how it breaks them down. Because you expect the red blood cells to be containing hemoglobin, so it must also be broken down into its constituents that will later combine in the new red blood cells that will have been formed. Also, the liver synthesizes plasma proteins from amino acids. That is the albumin, a transport molecule, globulin, a transport molecule of hormones, and prothrombin and fibrinogen. These are the clotting factors. They are involved in the process of blood clotting by the platelets. The liver also produces bile, which is a mixture of salts and cholesterol. Bile emulsifies fats during digestion in the duodenum. The reason why we don't have digestion of fats in the mouth and the stomach is because there is no bile that would emulsify fats that can be easily digested. The liver also stores fat-soluble vitamins A, D, E, K, and water-soluble vitamins B12 and C. It also stores minerals like iron, potassium, copper, zinc, and trace elements. Also, the liver stores up to 1,500 centimeters cubed of blood in its vast network of blood vessels, hence acting as a blood reservoir during very emergency cases. Another role it performs, it destroys all hormones after exerting their effects in the body. Now, these guys have to be removed. Once they have performed the role that they were supposed to do, they should not be allowed to stay. Because once they stay, then they will bring about the effect that we know called the positive feedback mechanism. Because if, for example, insulin was produced to bring about conversion of excess glucose into glycogen, then glucose stores are now reducing. They have been restored back to the norm, and this guy is not inhibited. You expect the effect to be continuous. The liver also performs the role of detoxification, which is detoxifying the poisonous substances. That is, we are removing the poisonous nature from these substances. Naturally occurring substances or compounds absorbed by the body can be toxic if allowed to accumulate and are therefore rendered harmless by the hepatocytes. For example, ethanol is oxidized to ethanol, and other products of detoxification are usually excreted, but sometimes they are stored because they are not toxic. They have been converted from the toxic or the poisonous substances, and now the products are not toxic, so they can be stored in the body until they have the best way of being excreted out from the body. So basically, that gives us a highlight about the homeostatic roles of the liver, how it brings about deamination, detoxification, storing different substances, lipid metabolism, carbohydrate metabolism, that is maintaining blood glucose levels, and protein metabolism. Thank you. Thank you very much for watching and for listening. Have a good day.